Hello, everyone. Welcome back to the NGSeminars. We are very happy to start this seminar series again. And today we're here with Anamaria Elek, who will give us a talk titled Coral Cell Atlas and a Quest to Understand Cell Type Evolution. If you have any questions throughout the talk, just put down your comments or your questions on YouTube or Discord. On Discord, we have a channel called Coral Cell Atlas. So you should check it out and ask us questions, and we will answer them. Anamaria is a PhD student at the Center for Genomic Regulation in Barcelona, Spain, in the group of Arnau Sebepedros. She works on gene regulation and cell type evolution for early metazoans. On a day-to-day basis, this implies analyzing and interpreting single-cell transcriptomics and chromatin profiling data, focusing on transcription factor usage, motifs, and gene regulatory networks. So, Anamaria, the floor is yours.
Yeah, thanks, Gladys. Thanks for the nice introduction, and thanks for everyone in the NGS School organization team for giving me this opportunity and privilege to inaugurate the new season of NGS seminars, and many more interesting talks to come. And I'm really happy to be the first in line. So today, now in the next maybe an hour or so, I will tell you a little bit about our recently published research. As Gladys already said, it's the Coral Cell Atlas, and how we plan to use this to understand the evolution of cell types. So to start with, first thing is, why do we even study corals? One thing is that they are beautiful, if you didn't know before, you've seen now in my first slide, but they are also extremely ecologically important because coral reefs are real biodiversity hubs, and they're home of more than a quarter of all marine species. And in the recent decade or so, climate change-driven increase in temperature and increase in acidity of seawater have caused a repeated coral bleaching and subsequent death of corals. So this impact is so widespread that it has even been suggested that only climate change reversal can save coral reefs globally. But there have recent study published recently have been showing that, for example, in this study published in Science has shown that local changes, so such as reduction in overfishing or pollution, can actually act to mitigate them. And this is one reason why we do research in corals, is to help as they put it, the authors of this study nicely put it, is to help the coral reefs survive the Anthropocene. Despite their huge ecological importance, however, not many of the fundamental aspects of stony coral biology are still not known and not understood, namely, especially the cellular contact of the important molecular processes that give rise to coral reefs, and this is something that we focused in our work, shown here in the second picture, and we basically wanted to characterize molecular and cellular basis of the biology of stony corals, for one thing, and for another, we wanted to use these corals and their cell specifications to put them kind of in evolutionary context in the study, explain why we use corals for this. So with both of these aims in mind, we set out to do single-cell RNA sequencing and to kind of assemble the cell type atlas of one coral species. Now a little bit, trying to move my slide. Okay, now a little bit about single-cell RNA sequencing. So we've been also over the last decade or so, maybe less, we've been witnessing how the single-cell RNA sequencing has been nothing but revolutionizing for the study of cell types. And more than 1,000 single-cell transcriptomic studies, for example, have been published up to date. Majority of them, however, in the organs and tissues of the handful of vertebrate species, and majority of them collated in large atlas projects such as Human Cell Atlas or Tabula Muris or Fly Atlas, which is now in preparation. And for example, Human Cell Atlas has annotated over 40 million cells so far, and this is a tremendous achievement, and it has many exciting implications and applications of the single-cell RNA-seq. However, a striking pattern in all these growing trends is that more than 90% of the data sets actually come from human and mouse. And apart from the increasing number of data sets, what has been rapidly increasing as well is the throughput of single studies, shown here in this second plot, where now recently in this year and last year there have been single transcriptomic studies reporting over million of cells. So it's been really remarkable trends. However, as I mentioned earlier, apart from human and mouse and few other vertebrates and maybe Drosophila and C. elegans, relatively few other invertebrates have had their cell repertoire characterized. And you can see this annotated on the metazoan phylogenetic tree that I'm showing on the right, which lists different taxonomic groups for which we have like either genome or transcriptome available, and for which then we have or do not have, in most cases, any single-cell RNA data. So there has been a clear bias, but it's something that we are trying to work on, also in different projects. In my lab, we are particularly interested in this bottom part of this tree, which is the early branching non-bilaterian metazoans, and these include the phyla of ctenophores or comp jellies, porifera or sponges, placozoans, those are like flat animals, flat marine animals usually living at the bottom of the sea, and cnidarians, which include before mentioned corals. So we are interested in these animals because they are perfect models for studying the cell type evolution. Why? Because they are relatively small, they are amenable to whole organism sequencing, so we don't need to isolate individual tissues or organs, so we can characterize all the cell type that they have, and this is usually a smaller, a limited number of cell types than what you would expect in these other phyla. And importantly, because of their position on this metazoan phylogenetic tree, they are the animals that were earliest branching. So they were first or earliest evolving multicellular animals. And then that's why we want to focus on studying their cell types, also to kind of learn the logic and the regulatory mechanisms and the effector mechanism that gave rise to these, that caused the different cell types to actually appear. So when it comes to these four phyla that we are interested in, here I just try to summarize what single cell studies have been done in these species. And you can see that we have at least one, or most of the cases one, species from each phylum that have had their cell types characterized by single cell RNA. So first two studies in 2018 reported single cell atlas for one member of each of these phyla, and then it was followed by like two more sponges atlases and four more cnidarians, two of which have actually been published last year, and one of which is this our work that I will be talking about in the next hour or so. So, to introduce you to our main star, our model organism that we use in this study, Stylophora. It's called Stylophora pistillata, and this is a reef-building stony or scleractinian coral. So Stylophora, again, as a reminder, is a cnidarian. So this is a group of animals that is actually one of the sister groups to bilaterians, and bilaterians include all the animals that you would normally think of, like, including ourselves. So it's close relative of bilaterians. It has a very broad Indo-Pacific distribution. It's thriving in these oligotropic and subtropic seas, shown here in the map, and it has three major stages in its life cycle. So it starts as a free swimming larva stage, which then settles on the surface and forms a polyp. And this polyp is actually the stage that is depositing large quantities of exoskeleton, of calcium carbonate exoskeleton, and it actually is most important for building building up the entire coral reef. And this is how these four polyps form then the adult coral colony. So what we did is that another important thing, that these corals actually have within their cells, within part of their cells, have also algal endosymbiont, and this symbiosis is actually very of a high metabolical importance because it provides corals with photosynthetic substrates important for metabolically intensive process of building up the coral reefs, and this is also something that I will come on later in the talk. So we sequenced 16,000 cells from the adult coral colony, and we identified 11 or 12 major broad cell type classes in this data. So here, what I'm showing in the upper part is the 2D projections of single cells clustered and by similarity of their transcriptional profile and annotated by the expression of known marker genes as different cell types. And the colors actually correspond to the schematic of the coral shown below. So this is where each individual cell type is found in the coral. So we have, just to summarize quickly, we have the large population of, or two populations of gastrodermal cells, which are on the inner side of the coral body. Some of them are those cells that are hosting the algal endosymbiont that I mentioned earlier. Then there is epidermis, which is on the outer side of the coral, here light blue, and related calicoblast cells. These cells are the ones that actually produce this exoskeleton. They secrete these calcium carbonate complexes and build up the whole coral reef. Then we also identify cnidocytes, which are typical cells of all cnidarians. So these are the stinging cells that cnidarians use to kind of capture prey, and also digestive filaments, something like equivalent of gut, you could say. We also identified germline, a subset of germline sperm cells, and two types of the immune cells, which have not been previously found in any of the cnidarians. They have been suspected that they exist, but have not been identified before. And finally, we identified a broad range of gland and neuronal cell types, which all come from the same lineage in the adult species. So as I mentioned, all of these cell types are annotated and they are supported, their identity is supported by the expression of known marker genes shown here in the heat map, and again color-coded by the particular cell type. And then we did the same thing for the two other stages. So we sequenced 3.5 thousand cells from the swimming larvae stage, and you can see that here you have a wider range of the undifferentiated or precursor cells, and you have much less diversity in the neurosecretory lineage, also fewer neuronal and gland cell types, and also the polyp stage, which has wider, for example, has wider proportion of the calicoblasts than the other two, because this is the stage that is mostly responsible for building the exoskeleton and the coral reef. So now I will. So this was kind of the overview of the things that we discovered in our coral cell atlas, and now I would like to focus on a couple of particularly interesting examples that we went into more details and characterized more thoroughly. And the first of them is the skeleton-producing cells, or calicoblasts, which, as I mentioned before, can be found on the external. So they are external cells that surround this aboral region of the polyps, and in the cell they are also found in the cell lining the skeleton spines and gastrovascular canals in interconnecting the polyps in the adult colony, and we confirm their expression by in situ hybridization. So first thing that we quantified is that there is higher proportion of these cells in the polyp stage than in the adult stage, as I already mentioned earlier, and also the calicoblasts in polyp and adults have different roles. So while the calicoblasts in the polyp are enriched in different metabolic functions, so they express various transmembrane channel and transporters, the calicoblasts in adult have more epidermal-like role. So they express, or they are enriched for the elements of extracellular matrix, and so various fibronectins, lectins, caterins, and so on. So next thing that we wanted to characterize is where do these cells come from and what are they related to. So we know from before that that calicoblasts are of epidermal origin, but we wanted to we did a phylostratigraphic analysis to determine their evolutionary age. And so we are doing this by looking at genes that are specifically expressed in calicoblast and stratifying them by evolutionary age that they come from. And then we can see that that there is an enrichment in calicoblast of genes that originated in the hexacoloraria and scleractinia lineages, or the genes that are specific for Stylophora, both in adult polyps and in settling polyp calicoblasts. But however, the calicoblast-specific transcription factor, which are the transcription factors, are those proteins that we always assume are the most responsible one for the cell type identity. So there are 3 of transcription factors that are identified specifically in calicoblasts, and they have an earlier evolutionary origin. So they are like from holozoans or cnidarians or placozoan origin. So what these results suggest is that calicoblasts originated from epidermal cells during scleractinian evolutions, and in this process, actually, the ancestral transcription factors that were usually linked with epidermal function now recruited novel genes of younger origin to form a new specific cell type of calicoblasts. So that was the first important coral specialization that we looked at, and the next one was those cells that host endosymbiont algae. And this, as I already mentioned earlier, this stable photosynthesis with the dinoflagellate algae is crucial for building coral reefs, because it satisfies the high metabolic demands of this process. So we again identified these algal endosymbionts in the subset of coral cells that are found around the gastrovascular cavity and the gastrovascular canals by in situ hybridizations. And because they are the photosynthetic algae and they have the autofluorescence, we were able to look at them under the microscope, and also to devise a FACS sorting strategy to specifically isolate this subset of cells that contain endosymbionts. Then we quantified which coral cells are actually those that contain these dinoflagellates and the symbionts, and we found them to be a group of gastrodermal-like cells in green, which we then termed alga hosting cells, and which generally have around 50% of algal occupancy. So it means that around 50% of these cells would contain the endosymbiont. And then there was also another group which we termed mitotic host cells, which had higher algal occupancy, or with median around 80%, and they also express genes like nanos, tudors, multiple tubulins, and cyclins, and generally genes involved in the cell cycle. So that's why we term them as mitotic host cells. So we wanted to characterize also the transcriptional program of these cells that contain endosymbionts, and because they are originally gastrodermal cells, we compared them, so the algal hosting cells, to gastrodermal cells, and we found that the algal hosting cells are enriched for distinct metabolic functions. So they express genes involved in different metabolic processes and different transporters and different channels. And then we also, when we looked at the genes that are expressed in the endosymbiont in this algae, we found that the expression of these genes is depleted in this endosymbiont algae compared to the free living algae. So there is also a free living version that is not in the photosymbiosis with the coral, and this free living symbiont would express, or would have gene expression program enriched for these transporters. So now because this endosymbiont algae don't need them because they are expressed by the coral. So this is the basis of this photosymbiosis. And this is how this related to the fact that the algal symbiont will then transfer photosynthetic products to the coral, and coral will transfer the inorganic carbon to the endosymbiont, and everyone is happy and everyone benefits. So the next thing that we also wanted to do, same with calicoblasts, we wanted to look at the evolutionary origin of these cell types, and to do this, we compared these endosymbiont containing cells in Stylophora in our model coral with another symbiont containing coral that for which there was a single cell RNA data available, and this is Chenia. So we wanted to look whether this transcription program of the endosymbiont containing cells in Stylophora is more similar to the gastrodermis in Stylophora from which it originated, or it's more similar to the endosymbiont containing cell in Chenia and the other way around. And we actually found that that's not the case. We found that Stylophora host cells actually more closely resemble Stylophora gastrodermal cells than the Chenia host cells, and the other way around. And this actually then suggests that there was probably a kind of convergent evolution or kind of convergent mobilization of the same molecular pathways in the gastrodermal derived host cells in these two species. So they do share some similarities, but they are more similar to the respective gastrodermal cells. So that was the characterization of the endosymbiosis that we did, and one final kind of short story that we wanted to focus on were the immune cells. And as I mentioned before, it has been previously hypothesized that there is innate immunity in corals, and that it is an important component of this both of this symbiotic homeostasis relationship with the algae, and it's also important for response as a response mechanism for various environmental stressors and so on. And actually, in coral genomes, there have been many genes annotated with immune function. However, immune cells as such have not been previously characterized or identified in any of the cnidarians, and so not in the corals either. And in our single cell atlas, we identified two classes of immune cells, and both of them express the known immune transcription factors, NFAT, for example, and interferon regulatory factors 1 and 2, as well as various immune-related effector genes, such as genes coding for antimicrobial secreted proteins, transmembrane receptors, different signaling factors, also genes involved with apoptosis in the protein turnover, inflammatory response, and so on. And important aspect of this is that the involvement of these conserved transcription factors and orthologs, so they are conserved across animals, and this implies that the existence of evolutionary conserved transcriptional program for immune response across animals. And here one example that we showed is for the interleukin receptors and toll-like receptors in all the animals that are orthologous to, so that Stylophora orthologs, Stylophora proteins that are orthologs to the same proteins in other animal lineages. And this kind of it's very preliminary result, but it's something that I think we will be able to see and confirm with further more detailed and more targeted experiments. So these were kind of three short stories that we were particularly interested in characterizing related to that we could find in our single cell atlas. And now we also wanted to do a little bit more systematic comparison. So I told you earlier that we sequenced and analyzed 3 different stages of Stylophora, and first thing that we wanted to see is how the cell types from different stages compare to each other, and can we identify the kind of similarities in transcriptional programs across the different life or developmental stages of Stylophora. And to do this, we devised an approach in which we first selected, so we combined the expression data for all the 3 stages, and we subset it for almost 6,000 variable genes. And then we calculated Pearson correlations between individual transcriptional programs based on these variable genes. But we did it, and followed by hierarchical clustering, but we did it in an iterative approach. So with doing 1,000 time random downsampling of these variable genes and doing clustering then on this downsample data. and then looking at how many times or what is the proportion of the co-occurrences of different cell types in the resulting cluster. So why did we complicate it this much is because we didn't want to our general similarity matrix to be dominated by the abundant cell types with very strong expression signatures. So we wanted to give the chance of less abundant cell types, such as, if you remember, cnidocytes or immune cells, which are minority of cells, so they're smaller. We wanted to give them the chance to appear in these similarities. So we didn't want to kill these similarities in our matrix. And this is the result that we get if we plot it as a kind of heat map. And you can see that, so again, the color coding is the same and corresponds to cell types. And you can see that different neurons, for example, from different life stages cluster together very well, as well as gastrodermal cells, for example, and these cnidocytes, they also form one cluster. Gland cells less so, but there is a biological reason behind this, that gland cells are very diverse within themselves and they don't have a lot of unifying transcriptional, they don't have a strong unifying transcriptional signature, so they form maybe two smaller clusters. And then also epidermal cells and related calicoplates that I talked about earlier, they cluster together. But what you can see from this heat map is that, so this cell type similarity, that is a highly it's very hierarchical data. So you could call this, it's not there is no one level of clustering in this data that you can decide. So we thought that the better representation for this would be kind of building a cell type tree based directly on this matrix. And this is what I'm showing in the right side of the slide, where we put all these different cell types on a tree, and then for each, you can look at each node as which corresponds to each different level of clusterings, and see what genes are supporting the similarity between the descendant cell types in the tree. So the numbers of the nodes, the numbers shown on the nodes actually correspond to node support, which is the number or percentage of these iterations that in this percentage of iterations, these cell types cluster together. And on the side of the tree, there is a genes listed, so both transcription factors and important effector genes, marker genes that support all of these similarities. So we kind of were very happy with this, because we saw that this approach works quite well, and that we can retrieve similarities at different levels for different transcriptional programs. And then we wanted to actually take this one step further and use the same approach to compare cell types between different species. So as I mentioned in the introduction, there is 5 in total, but at the time of the writing of this paper, there were 3 more single cell atlases of the cnidarian species available. So apart from our data, there was a data on the Nematostella vactensis, which is sea anemone, like this kind of a model system of cnidarians. There was also another coral, Chenia, that I already mentioned earlier, because that this is also a coral that contains endosymbiont, but it is a soft coral, so it doesn't build the reefs. And there is also Hydra, which is a seawater hydrozoan animal, actually. So we wanted to compare the cell types between these 4 different species. To do this, we reanalyzed the data for 3 different species to have them processed in the same way and at the same level of granularity as our Stylophora data, and then did the same thing as we did for the cross stages cell type clustering, that we selected variable one-to-one orthologs in this case, and perform the same iterative procedure of clustering and downsampling and clustering again of the cell types. And this is the resulting tree that we get. So we did this at the level of broad cell types. So what I mean by this is that we were looking at all epidermal cells together, for example, and all neurons together, and all gland cells together, and so on, and like this in every species. And we were able to retrieve strong transcriptional similarity across these 4 species, where you can see that epidermal cells always cluster together together with the related calicoblast, which originate from epidermal cells, as well as digestive filaments for different species, germline cells like cnidocytes from all different species clustered together. And you can see, for example, cnidocytes from the three anthozoan species clustering together, and the cnidocytes from Hydra, which is most distant one, are also clustering as further apart. So these results made a lot of sense, and then again, as before, we could identify the responsible or the underlying transcription factors shown in this next to the nodes in this tree, which are giving identity to these individual cell types. So this was something that we were also happy with, but the next thing that we wanted to look at was to look at the cell types with a higher level of granularity. So we wanted to look, for example, at the individual neuronal subtypes or neuronal cell types, and we wanted to identify whether we can identify the corresponding more narrow sense cell types between different species. Now, to do this, we used a little bit different approach. So we used this another metric, not the correlation, but the metric called Kullback-Leibler divergence, which is a measure of how one distribution is different from another. And we actually hypothesized that this might be a good measure of diversity of very specific transcriptional programs. So not the broad ones, but the narrow sense cell types, because it doesn't require selecting variable genes, but it takes into account the entire distribution of the gene expressions across a cell type, and then compares it between different cell types. And what it does, it effectively quantifies the difference between these two distributions. And that means that unlike Pearson correlation, this similarity or dissimilarity will not be dominated by a subset of highly expressed genes. And then here I'm showing the results of this analysis. So here these are the cell types in Stylophora, for example, and this is Nematostella. And the links between individual cell types are the top 2% of top 2% most similar pairwise cell types between these species, with the width of the link corresponding kind of to similarity or being proportional to the similarity. And the thing that you can see here is that the broad cell type relationships are still holding. So there is, like, for example, gastrodermal cells in Stylophora are linked mostly to gastrodermal cells in Nematostella. Also, neurons mostly correspond to neurons. Gland cell mostly correspond to gland cell. However, in most of the cases, we cannot identify direct one-to-one correspondences between individual cell types. So these similarities are dominated by many-to-many relationships. There are some cases, like this one shown highlighted in the circles plot now, where one particular neuronal subtype in Stylophora corresponds to one particular neuron, most highly or significantly corresponds to only one neuronal subtypes in Nematostella. And then we can look for the genes, again, that are markers and specifically expressed in this cell type, in this kind of sister cell type, let's call them, and not in the other cell types. But for the most cases, and also for the two other species, we mostly see the many-to-many relationships. So why could this be, and why what do we think, why is this happening? So even though if you think about it, you're probably thinking, they are all cnidarians, there are some corals there or something, so they are very closely related. But in theory, these species, they are coming from 4 major cnidarian lineages, and they are separated by 500 million years of evolution. This is the equivalent time of when the land plants first evolved or separated from the water. So it's kind of very long evolutionary time that these species have been evolving separately. So what we want to do next is to expand our analysis with more closely related species. And now I'm showing here the updated tree so in bold are the species that we had in this study, and there are 3 more species added that are closely related to them, for which single cell data is or will be soon available. And we plan to then extend these analyses, and we hope that by comparing between more closely related species, we will be able to pinpoint similarities between individual cell types. And this is something that we want then reconstruct is the more detailed cell type tree, but not on the broad cell type level, but on the narrow cell type level, in which then we can, by comparing it to species tree shown here in this cartoon for this simplified example, when we have three different species, and when we create the cell type tree and we use the species tree to determine evolutionary relationship of these cell types in the cell type tree, so we can identify the divergence. So when this one cell type in one species diverge into different cell types or segregations, when some cell types involves different functions, or extinctions or losses of some cell types, or gains of some cell types. So this right now, with this resolution of data that we have, we cannot do, because the kind of cell type tree that I showed you on the broad cell type level doesn't work on the narrow cell type level, because they are dominated by many similarities. But it's something that we are hoping to do soon, now in the future. So that would kind of wrap it up, the 3 or 4 major analyses that we did in this paper. And the thing that I want to conclude with is that for all these analyses that I showed you so far, we included them in the interactive web applications. So we uploaded all our data in a Shiny app, which can be freely accessed and explored, because we want to make the data available for the community, for those people who are in the end working on the corals, on the coral biology, and who are working on studying and fighting for coral reefs and helping them survive the Anthropocene. So we included our clustering solutions for all the 3 life stages, with 2D projections and the heat maps with gene expressions in the app, when anyone can download this data, or also explore it interactively, so you can look at the expression of any of your favorite genes in there in the 2D projections or in the heat maps, download the sequence of the genes, also groups of genes, or also retrieve the most highly or more specifically expressed genes in any particular cell type. So this is especially useful for people designing experiments, like targeted experiments and in situs and so on, to find something that is characteristic of a particular cell type. And because all of this depends on the threshold that you select, we don't want to have people rely on the threshold sources that we chose for the paper. So you can choose your own fold change values, or your own percentage of cells expressing this gene, and so on, whatever it is most suitable for your applications, and download this data. And the same thing for the cell type comparisons. We included the interactive versions of both the cross stages and cross species tree, and you can explore any of this clustering at any of the levels, so at any of the nodes in the tree, retrieving the genes that support it, also with different stringency or different expression threshold, or with different orthologs supporting it. And we also provide the pairwise comparison between species on this narrow cell type level, where you can try out different metrics for comparison. So the KLD that we used in the paper, but also the correlations, the Jacquard distances, and so on, also with different sets of orthologs. We try to make this as transparent and as useful for the community as possible. And this has been very helpful for us in the interpreting the data and in the writing and analyzing the data for the paper. And we really hope to expand on this in the future, and we hope that this becomes the standard, not just for the human and mouse data, for which I'm sure people working on this have a wide variety of options available for how to retrieve and interactively export the data, but also in the non-model species community. It is something that I think will improve the research very, very much. So with that, I would like to wrap it up. And thanks first and foremost my lab, my supervisor, and the people mostly who have been working on this paper, so Xavi, Marta, and Simon. And also our collaborators in Israel who have done the most of the experimental work, starting from sampling the corals to sequencing and in situ and so on. And to collaborators in the UK also help with the experimental setups. And that's more or less it from me. I'm happy to answer any questions if some have popped up. I haven't been really keeping an eye on the Discord or YouTube.
Thank you very much, Anna. It's a lot of work. I can see that your team has done a lot of analysis with this data, and it's so interesting and very nice to see that we can really learn a lot from corals and their evolution from single-cell transcriptomic data. So congratulations and thank you for the talk. So I imagine that, I myself and maybe the audience too, we don't work with corals. So perhaps I can start with a question about them. What are the challenges did you find maybe when profiling the transcriptomics? Is it difficult to get the samples or to isolate the RNAs and stuff?
Yeah, spot on question. It is extremely difficult because most of these species, well, nowadays actually for this one, it is possible to grow it in the lab. But this one that we analyzed was sampled from the wild, and it was that was a funny story because they sampled it in the Mediterranean in Gulf of Elek in Israel, and the sequencing was actually done in Spain. So it included it was a crazy story of taking the samples in a car on a plane to Spain, coming in the evening, sequencing overnight because we didn't have the environment to keep them in the aquariums or so in the lab. So it had to be done like as soon as possible, and it was a monumental effort by the experimental team, and that's for one thing for sure. Then when it comes to protocols and so on, you can imagine that most of the protocols will need a lot of optimization. So most of the protocols are developed for mouse, human, or cell lines. And then when you're working with the whole organisms, that complicates things a lot. So also there a lot of modifications had to be done too, and a lot of troubleshooting. And again, massive credit to the people working on the bench on this. Then when it comes to the analysis, you are again facing a set of challenges when working with non-model species. Like you are usually working with genomes of less quality, so they are like usually thousands of scaffolds. You don't have chromosomes. Then gene models are not always that great, and gene annotation, which is usually you don't know what these genes are. Usually the annotations are assigned based on some orthologies, and this is so for many of the genes we don't know where they are, and then if you look in the paper, there are a lot of times when we just put the gene ID because it hasn't been annotated so far. For example, the gene in calicoblasts that we found is specifically enriched in calicoblasts, we don't know what it is. Well, but at least now we know it's specifically enriched in calicoblasts, so it's also improvement on that aspect. So yeah, there are many of the drawbacks, but there are many of the novel and exciting insight that we would just be missing out if nobody worked on these things.
Yeah.
It's worth the effort.
And that makes the result even more special, because of all the hard work to even get the samples. You mentioned that the coral reefs that in your study was from the Mediterranean area. Do you think that the ecosystem or the natural environment in that area changes the cellular level for these corals, and maybe it's different for corals from the Indo-Pacific that you mentioned?
Yeah, so this species is present in both Indo-Pacific and Mediterranean and so on. Now that would be interesting to see. It's for sure an interesting thing that would be to look at the single cell level whether there are major differences, because from the few studies that have been done so far, we can't really tell. No. The single cell level, whether we could do something like population genetics in corals, it would be very cool. And it has been done in a different, I think in one coral that I know of that has something similar to population genetics has been done, but for this one not really so far.
So do you think that building upon this results, do you think that you would collect more samples and do the same kind of analysis, or do you think we know pretty much enough about corals their evolution? What do you think?
I think we definitely need more samples, and we need more species, not samples but species specifically, because to make any reasonable evolutionary claims as I showed you before in the cell type trees, with these 4 species that are very distant, it's impossible to narrow it down to something more than very broad cell types, very broad level. But with the couple of more closely related species, some of which we now have in the lab, I think we will be able to tell much more. And then we also want to, when we get this resolution, like evolutionary resolution, we want to go into the regulatory mechanism behind all this and to identify what particularly, not just transcription factor, but the whole gene modules or gene regulatory networks, how are they used in different cell types, how are they driving the specification, why some species develop or keep some cell types, why some lost them, and so on. So I think we need more sequencing, but more species, not necessarily more samples, but more species.
All right. We have another question. They're asking, as a computational biologist, what exciting experiment you're hoping to do, or in the future, what to spark from this results? What are you hoping to see?
So yeah, so the thing that I was most excited to work on in this paper, and the thing that I'm most excited to work on in the future, is this whole comparative genomics aspect of the whole story. So it will, during my PhD, I think it will mostly be on cnidarians, so different cnidarian species, and comparing the transcriptional programs between different cnidarians. But then in the lab, we also have people working on these other phyla of the non-bilaterian metazoans. And then for me, computationally, the biggest challenge will be to identify the regulators, the transcription factors and their target effector genes, and to kind of assemble them in gene regulatory networks. We then compare the different cell types based on, not like now, I want to focus it more on the regulatory aspects, because I think this is what drives the cell type identity in the end. So for that, we will also we are planning to do also single cell attack integration, to kind of be able to narrow these pinpoint this regulatory relation more precisely, and let's see what comes out of it. But I think that's going to be very exciting.
Oh, we have one other question here that just came in. The immune cell types sound very interesting. What do you think they fulfill? What kind of roles do they fulfill? Are you able to see adaptation in the immune system because of the presence of algae? Because of the symbiosis?
Yeah, that's a very cool question, and something that we were trying to also tackle for this particular paper, also experimentally. But in the end, because the experiment didn't work, we couldn't kind of go on to characterize it in more details. So the things that we found in these immune cells were mostly things related to innate immunity, which has again been suspected that there should be innate immunity in coral, because one thing is this symbiosis with the algae, but another thing is the environment, because this is a colony and there are a lot of environmental stressors acting on these, or pressuring on these coral reefs, and then there is definitely, this is probably the major role of this broad innate immunity in corals. Again, we didn't go into details into looking in particular functions that may be taking place there, but it's I hope something that will be addressed in future. Also, because one of the reasons why I didn't work, because immune cells in corals, well, like in other animals, I suppose, are not localized like most of these cell types that we identified. Like they are not in one particular place. So we found them. That's why also we didn't have in situs for them. We found them all over the place. And then so something that has been raised, that the question was whether they are really a dedicated cell type or they're just kind of the reaction of the coral, or whether this is some other cell type which has been under stress and now it just changes its transcriptional program to express more of these immune-related genes. However, we did several analyses trying to disprove this, because when you strip out the immune signature of these cells or these cell types, immune cell types, and you look whether they resemble in any way anything that's there, you're not left with much. Basically, you strip the cells of most of their expression to start with, and the signature that's left doesn't resemble any particular cell type. So this is kind of how in silico we kind of try to confirm this identity of immune cells, but the experimental part didn't really work out. So I hope this is something that, if not by us, that somebody else will look into this. I'm pretty sure they will, because we know people who are working particularly on immune cells in coral. So I hope that for them this will be a good thing and a help.
Yeah, it sounds very interesting, even for me who's not working on it. So good luck very much for your team and for the whole group just to do the analysis and the experiments. Thank you very much for your time. It's an interesting talk, and thank you for answering the questions too.
Thank you for giving me the chance to talk about this.
All right. So for everyone, the recording of this seminar, it will be available on our YouTube channel very soon. So feel free to check back to rewatch it. And a few announcements before we end. So first of all, we want to introduce you to our new event this year. It's called NGS Sprint. We will challenge participants to do group projects that they have to finish in a week. So that's why we call it a sprint. We have various exciting topics for each group. For example, transcriptomics, epigenetics, machine learning, multi-omics integration. So there's something for everyone. This event will be in September, and the details are on our website. We're going to open the registration soon, so keep an eye on that. And if you have any questions, feel free to reach out to us on Discord or through our email address. And then the second announcement is the next seminar. It's next month. Please join us for a very interesting talk about the telomere to telomere T2T consortium, which has done tremendous work in completing the human genome sequence. This year, they published a preprint describing their efforts, and we are honored that the lead author, Dr. Sergey Nurk from the NIH, will come and join us to share their work. So check out our website for the details, and see you all in July. Bye, guys.